we have guided almost 150 students under different programs, and this internship program is these are the second batch of students who have joined us. In the summer internship, we had only 5 students registered with us for the internship, but this time we have got good number of students, and all these students, I would like to tell you, are very enthusiastic of learning certain things, and that's why I and Yash actually hunted around and were searching for some speakers, and we really got very good speakers from all over India. You are one of them. I am welcoming you to this particular event, and would ask Yash to introduce how his journey into the venture, and then introduce you, and we will carry on forward. Okay, madam?
Thank you so much. It's okay.
Yash, please introduce madam, and then we will carry on with.
Okay, ma'am. So, good evening, everyone. On behalf of Kaustav's Careerica, it is my privilege and honor to welcome you all to this expert lecture on biotechnology and diagnostics, which is organized under the Module 2, Translational Biotechnology and Healthcare Innovation of the Winter Internship Program. I, Yash Nigam, serving as a brand ambassador at Kaustav's Careerica, and I am currently pursuing my Master's in Biomedical Sciences from Dr. B.R. Ambedkar Center for Biomedical Research, ACBR, University of Delhi. It is truly encouraging to see much enthusiastic participation from students who are keen to explore the translational dimensions of biotechnology. To begin with, let me briefly introduce Kaustav's Careerica. It is a flagship initiative under its parent organization, Prakriti Private Limited. The Kaustav Careerica was founded by Dr. Pratima Tripathi with a clear and powerful vision, that is, to bridge the gap between the academic knowledge and real-world scientific application. In today's rapidly evolving life sciences landscape, we strongly believe that conceptual understanding must be complemented by industry exposure, research orientation, and career clarity. Our mission is to mentor, guide, and empower students by providing structured learning pathways, hands-on exposure, expert mentorship, and career-focused programs. Over the period, Kaustav's Careerica has expanded its service to include internship programs, expert lecture series, research mentorship, skill development workshops, career counseling, and academic guidance that is tailored for the undergraduate, postgraduate, and research aspirants. Our long-term goal is to build a robust ecosystem where students are not just learners, but future innovators, researchers, and leaders in the science and healthcare. Let me give some views on Winter Internship Program, which is carefully designed to expose the students to the emerging and interdisciplinary domains that define scientific research, science communication, modern biotechnology, healthcare innovation, and professionalism. This program follows a structured module-based approach, ensuring conceptual depth, translational relevance, and real-world applicability. The Module 2, under which today's lecture is being conducted, focuses on the translational biotechnology and healthcare innovation. And today's lecture is on the biotechnology on diagnostics, like how the biotechnology is used in the field of diagnostics. So the diagnostics forms the backbone of the modern medicine, from early disease detection and personalized healthcare to the epidemic surveillance and precision medicine. Biotechnology has revolutionized diagnostics by enabling sensitive, specific, and rapid detection platforms, and today's lecture aligns perfectly with this transformative journey. Now, it gives me immense pleasure to introduce our distinguished expert speaker for today's session, Dr. Veena Rani. So Dr. Rani is a highly accomplished researcher in the life sciences whose work beautifully bridges virology, molecular diagnostics, immunology, and translational research. She completed her PhD in virology from Jamia Ahmad University, New Delhi, where her doctoral research focused on the mapping conserved histidine residues in the envelope protein of the dengue virus, a study with profound implications for the antiviral therapeutics and diagnostic development. Academically, she holds an MSc in microbiology from Acharya Nagarjuna University, Andhra Pradesh, and a BSc in microbiology, chemistry, and computer applications from Osmania University, Hyderabad, reflecting her strong interdisciplinary foundation. She has also worked as a JRF on a DST-SIR project focused on the histidine switches in flaviviral envelope proteins, further strengthening her expertise in viral molecular mechanism. One of the most remarkable contributions of Dr. Rani's doctoral work was her pioneer use of viral-like particles, VLPs, as a diagnostic tool. Her research demonstrated that VLP-based systems offer enhanced sensitivity and specificity compared to the conventional methods, while simultaneously eliminating the biosafety risks associated with live virus handling. This innovation represents a significant advancement in safe, scalable, and translational diagnostic platform, especially for infectious disease like dengue. Currently, Dr. Rani serves as a research scientist on an ICMR-funded project where she applies cutting-edge molecular and multiomics approach to study metabolic immunopathology. Her ongoing research investigates high-fat, diet-induced dysregulation of myelopoiesis in murine bone marrow, integrating bulk RNA sequencing, plasma proteomics, and multiomics data analysis. This work provides critical insights into how metabolism influences immune cell development and disease progression. What truly distinguishes Dr. Rani is her dual expertise, spanning from VLP-based diagnostics in virology to the multiomics-driven hematopoiesis and immune regulation. Her research exemplifies translational science where fundamental molecular insights are directly linked to the diagnostic and therapeutic innovation. Dr. Rani's research interests include virology, VLP-based diagnostics and therapeutics, metabolic regulation of the immune function, multiomics hematopoiesis, and translational application for immune-mediated and hematological diseases. Her journey is an inspiring example for the young scientists who aspire to build careers at the interface of the basic research and clinical impact. We are truly honored to have Dr. Veena Rani with us today, and I'm confident that her insights will not only deepen your understandings of biotechnology, but also inspire you to think critically about research-driven innovation and real-world application. I encourage all participants to be attentive, curious, and interactive during the session and make the most of this invaluable learning opportunity. With that, I now invite Dr. Rani to kindly take over the session. Thank you, and I wish you all an insightful and enriching lecture ahead. Over to you, ma'am.
Thank you, Ash. Thank you for giving a brief description about me. It's so encouraging. So I will share my screen. Is my screen visible?
Yes, ma'am, it is visible.
Yeah. Good evening, everybody. So today we will start this a small lecture, giving you the insights of how these modern diagnostics techniques have been developed in this biotechnology. So before we come into this lecture, we should know what are diagnostics. When these diagnostics comes into role. So what is the outcome of these diagnostics? Why they are so important in the medical field? So when a patient comes to the doctor with some symptoms or with some injury or with some diseased condition, what the doctor does or a medical expert does, he need to validate with this his condition with some clinical findings. So he has to validate it. Suppose without a validation or without any proof or without any recognizing anything that is wrong with that person, the doctor or a physician cannot give an right treatment to it. So here, nothing but this is diagnostics are nothing but testing and processing used by in the medical field to identify what the pathogen or what is the reasons for this condition. So it is nothing but recognizing. So here there are two statements I have mentioned. So one person says that she was given wrong medication due to an erroneous diagnosis. What does it mean? Why did she receive a wrong medication? Because she had the diagnostic report was an erroneous one, means it is a wrong one. That's why that is the reason for giving her a wrong medication. No doctor in this world will give a wrong medication. Why this happened? Because there was an error in the diagnosis. Then the second statement says that the doctor treated the patient with pneumonia with a correct diagnostic report. Why the treatment was correct here? Why did the doctor was able to treat the patient for the sake of with the condition identifying that this is pneumonia? because the diagnostic report helped the doctor to treat this patient correctly. So by this, now we understand how important is this field of diagnostics in the medical field. So without this, there is a saying that medical field is blind without diagnostics. Okay. So now from the past, when we see to the present, is my slide visible? Is it changed?
Yes, ma'am. Yes, ma'am.
Yes, ma'am. Okay. So now, when we see these diagnostics as a period of time, in the like from the past certain years to now, there is lot of improvement, right? The techniques that which we are using earlier are nowhere now. So what? Just I give you a recap how we will understand how this field is emerging in biotechnology, how fast it is growing, how blessed or how privileged we are right now, and what are the reasons for those privileges that we are enjoying now. And we will just have a recap how the diagnostic techniques were there in the earlier, and what are the diagnostics techniques that we are using at present, and what made this change happen. So in the conventional method, it means the past, in the past eras, in the past decades, there was no proper there are only, the doctor used to treat the patient only by certain physical examination, or else he used to he has only very limited instrumentation like stethoscope, like thermometer, and pH monitor, and only they completely relied on only microbial techniques. So like microbial techniques, if there is an infection in the body, they used to take the complete CBC count, and by counting the leukocytes, like counting the granulocytes, then the person is diagnosed that he has certain infection. But there is no proper thing what infection that is. So, and if there is some infection, only urine analysis and very limited biochemical experiments were present in the past to identify the or treat or diagnose any diseased condition. So because of this, what was happening when they had these limited most of them are microbiological techniques only, like they were using. So what happened with that? So there was very low specificity, and it is very less sensitive, and most of the results were giving false positive. And it is totally, all the results that was given by these diagnostic centers in those days were totally dependent on the operator. So if the operator does in a right manner, if there is an error done by any operator knowingly or unknowingly, there are different kinds of different result that was coming. That was nothing but false results. So even they were not unable to detect. Nowadays, if they even before developing the symptoms, symptoms also, we could identify there is some diseased condition in the patient. But in the earlier days, they were unable to detect any disease condition in the patient. So what was happening with that? So
All of you, please wait. I think Madam is having some network issue. She will rejoin. Okay. Okay, dear students, all of you are present here.
Yes, ma'am.
Eman, Imran, Tashbi, Fari, Talat, Zainab, Gautam, all of you are there?
Yes,
Yes,
ma'am.
ma'am.
Very good. Chalo, till ma'am rejoins, we will have some conversation. How is your work modules going on? All of you have attended the quizzes? How many of you have attended the quizzes? Is how many have not? Bolo bhai.
Ma'am,
Ma'am, quiz number 3 and 4 is about to.
3 is about, you are about to attend quiz number 3. 1 and 2 you have attended? Ayman.
Yes, ma'am.
Acha, Gautam. Okay, 1 and 2 you have attended. You have texted me also. Imran and others?
Yes, ma'am. 1, 2 completed है, ma'am.
3 क्यों नहीं complete हुआ बेटा? When modules have been completed, you should have completed quiz number 3 also.
Yes, ma'am. आज हो जाएगा.
आज हो जाएगा. ठीक है, Ayman. What about you? Ayman, you are there?
yes, ma'am. मैम अभी क्विज लेफ्ट है मैं आज मैं कर
आज कर लीजिएगा. All of you complete the quizzes because tomorrow I may check it, and then Monday probably you will have a class with me where we will complete module 4, and along with this lecture 2 more lectures are there. Okay, 3 more lectures are there. 15 को है, then 12th को है और 19th को है. So जो 12th और 19th वाला है, that are the lectures on entrepreneurship and innovation. वो जरूर attend करिएगा. Attend सभी करो, लेकिन उसको ध्यान से सुनिएगा so that you will get an idea how translational biotechnology can lead you to innovation and entrepreneurship. मतलब फ्यूचर में चलकर as a researcher come entrepreneurship में अगर आप enter करना चाहते हो तो what all you should keep in mind. And Dr. Shiv Kiran sir, जिनका lecture है, he has already been an expert in our previous programs also, so we know that he's a good orator. Orator का मतलब जो बोलता है. He's a good orator और बहुत अच्छी उनकी understanding भी है. He has also been conferred with Innovators Award. So innovation and entrepreneurship में Government of India के साथ he is working, and he is actually a very good person और जिनसे आप बहुत सारे questions भी पूछ सकते हैं. And he is very interactive with students क्योंकि उनका काम ही है students में awareness generate करना, how they can shift their research and academics to innovation and entrepreneurship. Okay. यश एक बार मैम से पूछ लो कितना टाइम लगेगा तो वी कैन कंटिन्यू विथ, मतलब मैं बच्चों के साथ कंटिन्यू कर सकती हूँ जब तक वो ज्वाइन करेंगे. एक बार इंक्वायर कर लो बेटा उनसे.
ओके मैम.
और आप लोग जब पढ़ रहे हो मॉड्यूल्स तो डू यू हैव एनी इश्यू विद द मॉड्यूल्स? अच्छा, एक ऑनेस्ट सा फीडबैक लेना चाहती हूँ. आई वांट टू टेक अ ऑनेस्ट फीडबैक. डोंट हेजिटेट व्हेन आई एम आस्किंग. Be very frank in answering the question that how are you liking the internship? What is your opinion about the internship? Are you liking it or you want something else to be done into it? अभी बता दीजिए because you still have some 15 days time. तो 15 मतलब हाँ 10-12 दिन समय है तो we can incorporate those things also. Otherwise overall आपका opinion क्या है about the internship? One by one. Ayman, don't hesitate to use this particular forum. हमेशा क्लास में प्रोएक्टिव होना चाहिए.
I am understanding it and enjoying it also.
Okay. You don't want to add anything into it, मतलब कि आपको नहीं लगता कि there should be something else that you want to learn, or this much is actually more than enough for you?
Yes, ma'am. I am getting to know many new things, so this is enough and good for me.
Okay. So you were expecting this type of internship means when you were looking around for an online internship, right?
Yes, ma'am. I have got to know many things.
Okay. Imran, what is your opinion?
Yes, ma'am. मैम मुझे अभी तक बहुत अच्छा कुछ न्यूज़ सीखने को मिला है. और मैम इससे और मेरे नॉलेज में मतलब कि बढ़ावा हुआ है, मतलब और मुझे नॉलेज मिला है.
तो आपको जैसे इंटर्नशिप आप ढूंढ रहे थे या जैसे मतलब क्या एक्सपेक्ट कर रहे थे कि इस तरह की इंटर्नशिप मिलेगी, या ये इंटर्नशिप आपके एक्सपेक्टेशन के अकॉर्डिंग है, या कुछ और भी इसमें ऐड होना चाहिए? व्हाट डू यू थिंक?
यस मैम, सब सही है. जो मैंने एक्सपेक्ट किया था वही है, ऐसे ही.
अच्छा, आपने एक्सपेक्ट किया था कि ट्रांसलेशनल बायोटेक्नोलॉजी में आपको प्री क्लिनिकल स्टडीज वगैरह बताई जाएंगी? ये तो नहीं पता रहा होगा ना?
नो मैम, इतना नहीं।
अच्छा ठीक है, लेकिन इतना मैं आपको ये गारंटी कर सकती हूँ, आई कैन गिव यू द गारंटी दैट वॉट एवर यू आर लर्निंग हियर इज एक्चुअली गोइंग टू मीन्स हेल्प यू ड्रास्टिकली, बहुत अच्छे से ये आपके रिसर्च में फर्दर काम करेगा। एंड व्हेन यू विल बी स्टडिंग इन योर further graduation and post graduation and go for higher studies, you will feel that ये जो आपका background जो आपकी बन गई है, that is going to help you a lot as a researcher and as an academician, and will give you actually a door, it will open a door to think कि कैसे आप अपने अभी प्रेजेंट नॉलेज को फ्यूचर में यूज कर सको। और अब आप जब भी स्टडीज करोगे तो आपको लगेगा when your mind will become that much active कि वो सोच पाए कि इस पढ़ाई को हम कैसे यूज कर पाए। Tashmi, what is your opinion? Tashmi,
Yes, ma'am.
आपका ओपिनियन क्या है इंटर्नशिप के बारे में? आर यू सेटिस्फाइड विथ इट और यू वांट टू लर्न समथिंग एल्स? बहुत ऑनेस्ट सा आंसर देना।
Yes, ma'am. मैम अच्छी है मैम इंटर्नशिप,
अच्छी है।
नॉलेजेबल नॉलेज, यस मैम, नॉलेज मिल रहा है काफी कुछ, अच्छा है मैम।
अच्छा ठीक है, यश एक बार आप इन लोगों से पर्सनल ओपिनियन लो, मैं डॉक्टर वीना को कॉल लगाती हूँ। यश, यू आर देयर? यश, nobody is responding only. Okay, no issue children, we will continue. अगर कोई रिस्पॉन्ड नहीं करेगा तो I will take your class since you all have joined. ठीक है, will it work? अगर मैं मॉड्यूल 4 कंटिन्यू कर दूं अगर कोई रिस्पॉन्ड नहीं करता है तो?
यस मैम, नो।
गौतम,
यस मैम।
तुम ये साइंस ना बोलो।
हाँ जी मैम।
ओपिनियन अबाउट इंटर्नशिप में पूछ रही हूँ क्या? हाँ जी मैम।
मैम ऑनेस्टली जो जितना मेरे को समझना था, बेसिकली मेरे को बायोटेक का इतना नॉलेज नहीं था, तो मेरे लिए तो ये काफी ज्यादा बेनिफिशियल है। बट अगर मेरे को एक चीज मेरे को ये समझनी है कि अगर मैं मोस्टली मेरे को पता चलता था इट इज ऑनलाइन इंटर्नशिप, तो ऑब्वियसली थोड़ा थियोरेटिकल वे में ज्यादा अच्छे से समझने का मिलेगा। बट अगर हो पाता, मुझे आई एम नॉट कंप्लेनिंग और एनी डूइंग एनीथिंग, आई एम सॉरी फॉर दैट, बट इफ अगर इसको अगर प्रैक्टिकलिटी में एप्लीकेशन यूज करने को मिलता तो मे बी और अच्छे से मे बी हमारी एनहांसमेंट हो सकती है।
अच्छा, मतलब यू वांट सम प्रैक्टिकल नॉलेज आल्सो कि मतलब कैसे इसको अप्लाई करें या क्या करें, है ना?
हाँ, मतलब जैसे अभी हमने पढ़ा है, जैसे मैंने अपना टॉपिक, जैसे मैंने कार्टी थेरेपी यूज सोचा, बाकी ने जैसे mRNA वैक्सीन का किया है, तो इफ अगर हम उसको और अच्छे से अंडरस्टैंड करना चाहते हैं बाय प्रैक्टिकल और एक्सपेरिमेंटल थिंग, तो उससे मे बी हमारे जो प्रोजेक्ट है, रिपोर्ट है, वो थोड़ा और अच्छे से एनहांस हो पाएगा, हमारी साथ में नॉलेज और गेन हो पाए।
अच्छा, तो अगर आपको कैसा लगेगा इफ आई इंट्रोड्यूस सम वीडियो क्लासेस एंड ऑल, मतलब कि वीडियो बेस्ड क्लासेस अगर मैं एंटर करूं? देखो हैंड्स ऑन तो डेफिनेटली ऑनलाइन नहीं हो सकता ना कि आपको मैं लैब में सेल्स वो कराऊं, ऐसा तो नहीं हो सकता। बट
हाँ, नहीं, वो तो नहीं हो सकता।
हाँ, बट व्हाट आई कैन डेफिनेटली डू इज कि आप लोग जो प्रोजेक्ट जो आप लोगों को मैंने दिया हुआ है एंड व्हाट एवर यू पीपल हैव चोजन, तो मैं उसको एक बार वीडियोस के थ्रू या किसी सिमुलेशन की वीडियोस के थ्रू मैं उसको आपको एक्सप्लेन करने की एक बार कोशिश करूं। विल इट वर्क?
या मैम, इट विल वर्क।
हाँ, मतलब मैं यही पूछना चाह रही हूँ, आप सब ओके कर रहे हो, लेकिन दिस इज व्हाट आई वांटेड टू नो कि और क्या चीज इसको और क्या बेहतर कर सकते हैं कि मतलब आपको एक सेटिस्फैक्शन हो कि हमने 1 महीने में सच में ऐसा सीखा है कि वो चीजें हमारे आगे काम करेंगी, है ना?
क्योंकि ऐसे अगर मैं एक नॉर्मल सिंपल एग्जांपल भी लूँ, अगर उसमें देखा, अगर डिसएडवांटेज चेक कर रहे हैं उसमें, कोई हम जैसे रिसर्च कर रहे हैं उसमें हमें डिसएडवांटेज पढ़ रहे हैं कि इतनी रेंज है या फिर कहते हैं इतने हेल्थ के लिए वो यूजफुल है कि नहीं है, बट उसका मैं प्रैक्टिकल एग्जाम्पल कैसे मैं प्रूफ वो होना चाहिए ना उसको समझने के लिए, जैसे वीडियो सिमुलेशन होगा तो उससे फिर भी समझ आ जाएगा कि हाँ इतना कहीं से हुआ है तो पता लग पा रहा है।
ठीक है, मैं कोशिश करूंगी कि मुझे ऐसे कहीं से चीजें मिल जाए, या मैं अपने एक्सपर्ट से भी बात करूंगी, अगर वो लोग कुछ आपको एक्सप्लेन कर पाए तो बेहतर होगा। लेकिन डेफिनेटली ऑनलाइन इंटर्नशिप में अगर आप ये चाहो कि आपको प्रैक्टिकल लैब वाली फीलिंग दिलाए, तो प्रैक्टिकल लैब वाली फीलिंग भी अच्छे से अच्छे लैब में नहीं दिलाएंगे। बीइंग अ ग्रेजुएट या पोस्ट ग्रेजुएट स्टूडेंट्स को एकदम से प्रैक्टिकल नहीं करा पाएंगे, क्योंकि कार्टी सेल्स हुए या एमआरएनए वैक्सीनेशन की बात हुई या एमआरएनए इंजीनियरिंग की बात हुई, तो बायो इंजीनियरिंग की बात हुई, तो ये सब चीजें एक दिन में या एक महीने में सीखे जाने वाली बातें नहीं है। ये एक साल साल लग जाते हैं लोगों को सीखते हुए, एंड नोबडी इन देयर लेबोरेटरी विल अलाउ यंगस्टर्स और फ्रेशर्स टू हैंडल द थिंग्स। बट डेफिनेटली सिमुलेशन वीडियोस या वीडियोस अगर मिलेंगे तो आई विल ट्राई टू एक्सप्लेन ऑल दीज थिंग्स इन योर क्लासेस। विल इट वर्क, राइट?
Yes, ma'am.
Okay, Zainab, what is your opinion?
मैम ऑनेस्टली जो गौतम ने बोला है वही मतलब मेरा भी है कि ओवरऑल सारी चीजें अच्छी हैं और सीखने को भी बहुत कुछ मिल रहा है और अभी बहुत कुछ सीखना भी है।
हाँ, मतलब
लेकिन जैसा गौतम ने बोला है कि थोड़ा प्रैक्टिकल रहता तो
मतलब
थोड़ा
मैं
ज्यादा
भी
समझ
समझ
आता।
पा रही हूँ कि एक होता है कि जैसे चैप्टर लिखा हुआ है तो चैप्टर पढ़ के जितनी चीजें समझ में आती है, वीडियो देखकर उससे ज्यादा चीजें एक बार ग्रास्प हो जाती हैं, है ना? मतलब अगर मैं एमआरएनए आपको समझा रही हूँ तो पूरा का पूरा चैप्टर अगर समझा दूंगी तो आपको समझ में तो आ जाएगा, लेकिन अगर एमआरएनए के वीडियोस में आपको दिखाती हूँ या सिमुलेशन करके दिखाती हूँ तो दैट विल डेफिनेटली ब्रिंग अनदर इम्पैक्ट इन योर माइंड. This is what you people want to say, right?
यस मैम।
Okay. Talat, what is your opinion?
I think I'm quite satisfied with it, but I also think some video lectures are also
Okay.
very helpful for us.
That anyhow I was thinking, but आप लोगों ने, चूंकि मैं कंसर्न आप लोग का भी जानना चाह रही थी कि वेदर, देखो कोई भी अगर बच्चा सीरियसली पढ़ रहा है तो उसको डेफिनेटली कुछ कंसर्न्स होंगे. आई यूटिलाइज्ड दिस पर्टिकुलर अपॉर्चुनिटी टू नो योर ओपिनियन. मैंने पहले भी ये सोचा था कि आपसे मैं एक बार पूछूंगी कि मिड वे ऑफ द इंटर्नशिप में कि क्या है आपका ओपिनियन, कैसा जा रही चीजें, कैसा लग रहा है आपको, क्या आपको कुछ नई चीज चाहिए. Good that you people aligned with my thought and you said that okay, ये होना चाहिए. Even I was thinking about it and I will definitely try to introduce certain videos. मैं कुछ अपलोड भी कर दूंगी वीडियोस आपके LMS पर, जहां से आप आराम से अपने टाइम के अकॉर्डिंग देख सकते हो. ठीक है?
Yes, ma'am.
Okay, children.
यस मैम।
मैं कुछ अच्छे वीडियोस और सिमुलेशन के भी वीडियोस मिलेंगे तो मैं उस पर अपलोड भी कर दूंगी, जिससे आप हमेशा वापस जाकर उसको देख पाओ, बिकॉज LMS विल बी ऑलवेज ओपन फॉर यू जब भी आप चाहें. बस इतना सा ध्यान रखिएगा कि नोट्स को सर्कुलेट मत करिएगा कहीं भी. वो आपके लिए है, आपने उसके लिए पे किया है, आपने उसके लिए 1 महीने टाइम एनर्जी सब स्पेंड किया है, तो कीप इट confidential to yourself as well as respect the dignity of the venture also. And you can always do the surfing of the notes and the videos for your personal use, but not for others use. बाकी किसी को चाहिए तो they should either take permission or they should try to utilize other notes available on internet, not the ventures notes. ठीक है?
Yes, ma'am.
चलो, I think यश, are you there?
मैम
क्या हुआ मैडम का? आज का तो टाइम ही ऐसे हो गया. रुक जाओ एक मिनट. हेलो यश, वो तो फोन ही नहीं पिक कर रही है. हेलो
Yes.
बेटा, वो मैडम को कुछ ड्रास्टिक प्रॉब्लम हो गई है, तो मैं आप लोगों को मॉड्यूल 4 कंटिन्यू कर दूं, पढ़ोगे? Because you people are already there and connected to me, so shall I continue with module 4 if you want to? एक बार आप लोग अपना ओपिनियन बताओ, फिर आगे बढ़ते हैं। नहीं करना चाहोगे तो मैं फोर्सफुली तो नहीं पढ़ा पाऊंगी ना।
मैम मॉड्यूल 3 में मेरे को एक टॉपिक पे थोड़ा डाउट था।
पूछ लीजिए, यू ऑल कैन आस्क योर डाउट्स आल्सो।
मैम एक टॉपिक है इंटीग्रेशन ऑफ फार्माकोडायनामिक्स एंड फार्माकोकाइनेटिक्स, तो उसके क्विज 3 में कुछ क्वेश्चंस थे उस टॉपिक पे।
किसमें क्विज में थे?
जी, को इतना उस पे डाउट आ रहा था, वो समझ नहीं आ रहा था कि वो क्वेश्चन
अच्छा
क्या
ठीक है,
है।
रुक जाओ मैं ओपन करूं। क्वेश्चन नंबर, व्हिच क्वेश्चन आर यू सेइंग?
मैम
हाँ हाँ
शी इज जॉइनिंग अगेन।
हाँ, तो लेट हर जॉइन। वेलकम हर अगेन, आई विल वेलकम हर, लेट हर जॉइन। Hello, madam.
Hello, Pratima. I'm so sorry. I was keep on giving the PPT, and I was
No issues, ma'am. No issues. You can continue with the students. They were discussing their internship doubts with me, and we were actually engaged with each other. So you can continue, ma'am.
okay.
Welcome back.
So, do you want me to give from the first? Where did
No,
I?
you continue
Ma'am,
from where you left, ma'am.
from the second slide, ma'am.
Oh, okay. Acha, from here.
Yes, ma'am.
Okay. Oh, I was about to finish my. Sorry, thank you. So it's so sorry. I got a call. I thought it was some other call. I couldn't. So
It's
so
okay,
sorry
ma'am. It's
from my
okay.
side.
Okay. Continue.
Okay. So, acha, from convention. So from the convention, in the conventional methods era, in the earlier eras, in the past decades, we had only limited clinical examination methods. So there are very limited methods, like the doctor used to, when the patient comes, the doctor used to just check with his minimum instrumentation that had with him, like stethoscope, thermometer, and his BP monitor, and he after just doing some basic physical examination, he used to prescribe him certain drugs or else certain antibiotics by depending upon only these limited laboratory tests, like doing, if there is an infection, to check the leukocyte count in the blood, like doing the complete blood count, like urine analysis, some certain biochemical reaction assays. All these things are only available during those periods. This lead to a lot of false positive and false negative results, and they are very less specific, and it totally depend upon the operator, who he operated it, and based on the error or any error done by the operator was totally influencing the diagnostic results. So these is the limitations that the people earlier past eras experienced. And now, and in this case, there is no early detection of any diseased condition. If nowadays, if there is any disease, the person is about to become face any diseases, in past days, there is no early detection of any disease. And also the recovery period is so slow because of these lack of certain high throughput diagnostic techniques. And there was limited understanding of this disease mechanism. What is the causative of this disease, whether it is pathogen or whether it is cancer or whatever it is. There are very limited understanding of this disease mechanism, and because of this lack, the person was there in the hospital for so many days, and there is high cost in the treatment, high cost in the testings, and also in hospitalization. These are all the challenges the earlier people faced and the society faced before development of this diagnostic field. So Now from conventional, in the conventional method, as I said, many of them, most of the techniques are microbiological based only. The blood culture will be grown on the agar plates, and when to identify the strain, which pathogen is harming, is the reason for this infection, they used to depend totally on an automated instrument which is called as Vitech, and it is very slow process. And when come to the modern methods, the modernization of these diagnostics was totally after the discovery of the PCR polymerase chain reaction. So this played a vital role in this diagnostic field, where within a day the patient was able to diagnose what kind of to identify the DNA, to amplify it, and identify what kind of strain or what is the reason for his disease. So when we also know during a pandemic time, the COVID completely, the diagnosis of COVID completely depended on this qPCR based on, and also MALDI toffee based on the protein detection, and even the whole genome sequencing, these DNA micro arrays are also widely used in these modern methods. So if there is any antibiotic resistance that is present, we can do the whole genome sequencing of that pathogen, and we can identify the specific targets, the drug targets, and all these techniques we will we will see these techniques. I will explain these techniques, how this helped this diagnostic field to take it further. We will see it in the coming slides. After these modern methods, again the advanced methods, which are the biosensors, the POCTs, point of care testings, all these also revolutionized this biotechnology field, this diagnostic field very further. Like now we have the rapid COVID. During COVID, we have seen the rapid COVID testing kits, and also we have a pregnancy testing kits. We have the glucose monitor sticks that we use. All these things comes under these advanced methods. The patient, no need to go to the hospital. These are easily available in any pharmaceutical shop. They will buy it for themselves. They will analyze it, and they themselves can conclude what is the reason for their disease, for their symptoms. So this method totally revolutionized this advanced diagnostic field. So we will go one by one. What all the techniques that are widely used in this, because we are all master students. So just to encourage you, I am explaining these techniques, what are used in this modern diagnostics, which field revolutionized from convention coming into the modern methods. What helped to emerge this field much faster is nothing but PCR, as we explained, and also DNA sequencing. So the whole DNA sequence, whole genome sequence will be generated from these methods, and you can identify whether there is any mutation or whether there is any variation that is happening in these strains or else within the human. What is the reason for it? All these things can be done by this DNA sequencing, and also by getting this whole genome profiling, we can do a personalized drug selection. So you can check which drug has a target, can target to that particular gene by doing this genomic profiling, so that the person will be getting personalized drug selection, avoiding drug toxicity during cancer therapies. So even and also immunoassays are also widely used. And other than all these molecular biology techniques, these imaging technologies like CT scan, MRI, and PET scan, all these techniques which gives the 3D imaging of the particular condition, we have revolutionized and given. Particularly, it has focused. If a person have any tumor or a cancerous cell in certain organism, the 3D imaging will give clear images identifying and giving a contrast between the normal tissue and with these specific tissues, and given clear images of the patient, so the doctor can give his treatment in a very specific and targeted way. So this is the conventional, the difference, what all the difficulties, the conventional laboratory, what made this conventional laboratory so lethargic, so time-taking and so slow, and how these microfluidic POC devices, how these have fastened. So we can see here. So here in conventional method, so half of the time was taken up for the transportation, because there is no well-developed laboratory facilities in every area or in every region. So the sample is supposed to be transported, and then it has to be isolated. The nucleic acids have to be isolated from the blood samples, and then they are purified and they are amplified, and it takes around 12 to 72 hours. Almost it may take around 2 to 3 days. But when it comes to microfluidic, we have seen this is very evident in case of COVID-19. So in microfluidic, after development of this rapid COVID testing kit, what happened? Every person, they buy the kit for themselves, and they just a minute level of samples when we keep on the detector, so immediately they will be sensed and they will give the accurate results within few minutes. So this is how the difference, what is difference and the challenges, and how it was time taking when compared to the conventional laboratory methods and these microfluidic POC devices. So what made these molecular diagnostics? What are the techniques that revolutionized this field of diagnostics in biotechnology? So here these are the, as you are all master students, I don't need to explain this, but still I will explain you how they helped these diagnostics field to emerge much further. Is mostly PCR. After PCR invention only, these diagnostic field became fast, robust, and very specific in identifying the diseased condition, to giving the reports in a fast manner. So not only it identifies, it also finds if there is any mutation that is there. By these mutations, during COVID time, we are able to excuse me, we are able to identify the strain difference also, which strain it is and how this strain is varying, how regionally it is transmitted. All those things can be done by this PCR, the real-time PCR, which gives us the viral load, the CT value. Based on the CT value, we will identify the viral load of the patient, and we can give a proper right treatment, right, by diagnosing it properly, correctly. So with the doctor, it will help the physician to give the right medicine and right targeting the disease for treatment. So next is the next generation sequencing, NGS. We here in this case, we can get the entire genome mapping. It is a bit time consuming and bit costly, but still it will give the entire genome mapping of the samples. And by identifying the entire complete genome mapping, what happens? You can personalize your medicine. By this genome mapping, you will come to know what are the genes that are highly expressed, what are the genes that are low in expression. Based on that, you can personalize your medicine. And also you can edit in by this Cas9 gene editing method. You can also edit certain DNA strands, and you can also we can also give the treatment, a permanent treatment and a long-lasting treatment to the patient by editing certain gene sequences which are causative or else which are responsible for the disease condition. And microarray to analyze the gene expression, and we can also detect gene mutation, and also you can identify the biomarkers in various diseases. And blotting techniques like southern blot, northern blot, western blot to analyze the proteins and gene RNA and DNA, all those things are widely used in these modern diagnostics and also in molecular biology diagnostics. So this is the Cas9, how the person, as I see, my cursor just. See, she is the patient. She is it is written as Susan, and she is suffering from sickle cell anemia. What is the reason for sickle cell anemia? The reason there will be a mutation. Certain certain amino acids will be changed in that person. So this is the red color crooked line is the mutation that happened in his DNA double strand. And so to address this, to correct this, to edit this particular sequence, what was done? They have designed an guided RNA. This guided RNA, which is complementary to that specific sequence, was designed, and this guided RNA along with certain Casp9, it is an protease enzyme, caspase 9, they both are given to the patient. So what does this Casp9 does? It will chop off, or else it will identify. First, this guided RNA will identify it, identifying where the mutation is, and this Casp9, what it does? It will cut off the affected sequence, and it will again insert the guide RNA. What it will do? It will translate it, and it will insert the DNA sequence and replace the defected sequence with the right sequence, causing the person, giving a permanent remedy from her long-lasting disease. This way, how the very chronic diseases, cancer, and so many diseases where the treatment is not possible, all those diseases nowadays, it is a very new and emerging technique that is addressed for many diseases. So the complete genome sequence, suppose a person is about to develop the disease, even Yes, is it fine now?
Yes, ma'am.
So we are here, right?
Yes, ma'am. You can continue from NGS.
NGS. Okay, so after giving these samples to the NGS, so what they give us a list of genes. In this list of genes, we can know which genes are upregulated and which genes are downregulated. So, and based on these genes mapping to the proteins, we can identify. So, what all the person, what all the diseased, what this person can be exposed in the future to certain disease by overexpressing certain genes. What is the cause of this person to be faced that diseased condition? All the gene profiling can be identified by these NGS. So it's fine. I changed my slide.
Yes, ma'am, it changed.
Yes. Okay, so all these molecular, there are so many advantages are there in these molecular diagnostics when compared to our conventional, and also, so what all those advantages? They are highly specific and highly sensitive. There is very less chances of getting false positive results, and we can early, as I said, we can early recognize the disease by these whole genome sequencing, as I explained. So all the disease can be, before the symptoms emerge, we can identify whether this person, whether there is a chance of this person getting any cancer, or else whether this person developing any metabolic disorders. All these things can be identified by these NGS, by through NGS in early disease detection. And once you can also personalize when we give a whole genome sequence, those, if there is any mutation or there is any variation, or else we can identify with any gene variation which is specific to the drug target, so we can do a personalized medicine. So by this, what happening, by giving a specific, by knowing this information, which drug has a target to this gene variation, the person will not be exposed to n number of medication. So the drug toxicity will be reduced in case of this molecular diagnostics. And also it's a highly speedy, and also it gives an high throughput results, and we can detect any mutations by whole genome sequencing. And any like phenylketonuria, the person, we all know the person will have a difficulty in digesting phenylalanine amino acid. So by doing this genome sequencing, we can identify earlier itself by identifying any mutation in certain disease. So the person can be advised to certain dietary precautions, and the person can be made free from getting these disorders. And if there is any disorders that are assessed in this sequencing, we can also consult them to the genetic counselors and give certain planning so that they can avoid any risk in the future. So other than this molecular, and also even these immunodiagnostics also comes under this molecular only, but they all come under this molecular modern diagnostics. So this is also highly sensitive, and it detects the infection and any metabolic, any hormones, any biomarkers that are present in the diseased condition, like metabolic disorders, cancers, in any cardiac symptoms. So all these are specifically targeted by these antigen antibody reactions. It is highly sensitive. Even in micro or pico level biomarker present in the blood, or proteins that are present in the blood, can be highly specifically targeted by these immunodiagnostics. So this will enable to develop targeted treatment and also therapies. So what are the commonly used immunodiagnostic techniques? We all know as an MSc students, like immune precipitation. So here we identify any protein-protein interaction that happens. In chip assay, we will identify any DNA that binds to any proteins that are going to bind to that DNA target. So that what happens by this identifying these protein-protein complexes, and also immune complexes that are, or else any DNA, we can identify what happens. We can identify whether there are any transcriptional factors that are down-regulating, or else any upregulating these genes. By targeting that specific DNA sequence, we can target personalized medicines also to those by this technique. And also immunoassay techniques, like we all know ELISA, which is widely used during to identify any serological levels in the blood, This ELISA is widely used in viral infections and flow cytometry. So to assess any infection in the blood, there are specific markers in the blood for these cells like leukocytes. They have specific markers. B lymphocytes, they have specific markers. If the person is under infection, if there is any change in this blood profiling, all these can be done by this flow cytometry assay. And immunofluorescence is widely used for this cancer, for the biopsies and all, to check any biomarkers that are present in those tissues. So all those things will be targeted by this immunofluorescence. So by giving FICT antibody, we will address whether this particular tissue is expressing certain biomarkers like cancer biomarkers. All these things can be addressed by this immunofluorescence. And Western blotting, which is specifically by identifying any proteins within our samples. All these even these immunotechniques are also very widely used, robust, and also highly sensitive. And even a very, very minute of your sample or else the protein expression also can be identified by these immunodiagnostic techniques. So after this, we have seen molecular, we have seen immuno, and now the latestly used is AI-powered diagnostic system. So what is this AI-powered diagnostic system? So the diagnostic system, by integrating certain AI tools like machine learning, some techniques, some neuronal networking, all these things, by integrating them into diagnostic tools, techniques, they made this field still more robust. Even there is no need to nowadays, no need to go to the doctor. You can, when you are, there are so many techniques. They will insert some sensors in your body, some chips into your body. So the doctor itself will identify how your blood pressure is monitored. He will monitor from home itself. You need not go to the hospital. Suppose if he sees that there is some wrong with your blood pressure and all, with your sugar spikes, from there itself he will warn you, or he will try to give you the medication. So this made this highly robust, all these AI powered techniques. So among them, that is liquid biopsy, rapid pathogen identification, organ on chip, and disease outbreaking tracking. So we will check. My slides are visible to you?
Yes, ma'am.
Yes. So what is this liquid biopsy? So what happens in a cancer patient? So the cancerous cells will be undergoing apoptosis, and they will be releasing DNA into the blood. So based on by targeting this AI to this liquid biopsy, what happens? You can identify the circulating biomarkers that are present in your blood. So even at an early stage also, you can identify what are the biomarkers that are present in your blood, which should be at what level they should be. If they are above the required level, so that will be definitely identified by this AI based liquid biopsy, and the person will be treated, giving treatment at an early stages and in a personalized manner. So this is the advantage of liquid biopsy based AI powered diagnostic system. And rapid pathogen identification, this machine learning, what happens? It will identify, like in a group of crowd, you are identifying certain particular phase. So what happens? There is a program already given in the machine learning, and what it will detect, they have certain pattern recognizing factors. Based on that pattern recognizing factors, what it will do, this machine learning, this system will identify whether it is a virus, whether it is a bacteria, which strain it is. So that by doing this rapid pathogen identification, the person will be giving timely, you can identify. There is no need of the doctor or else the diagnostic to take some time to identify which strain it is. It directly say whether it is viral or what is leading to this diseased condition, the patient will be immediately addressed by identifying immediately the bacteria, or whether it is virus or whether it is fungi. So and the other one is organ on chip. So if there is any failure in your organ, like if there is in a heart patient, some of the valves can be damaged, or there is a failure in the liver, what happens here, based on this AI powered model, it can be your model can be developed, and it will, by developing these in vitro models, it will give it can make your treatment fast, and also it can be replaced. It can by targeting certain drugs to those diseased condition, it will correctly tell that which disease or which drug is suitable for your diseased condition. And also this disease outbreak tracking, what happens, it will monitors. So before the whole world, during COVID time, when we see the whole world was under this tracking, how the disease outbreak is happening. So we all know that there is going to be a lockdown after a certain period. Why? Because because of AI surveillance. What it will do, it will monitor the entire world, where the disease is propagating, how disease is spreading upon this public. So what will be the outcome, what will be the death, what will be the the symptomatic assessment, all will be done by this disease outbreak tracking. So this will help the AI based models have helped this diagnostic system to still evolve further. And now we are enjoying so many privileges, like so many kits we have, even our wristwatches. Even our watches which we wear, which will monitor our footsteps count, and it will monitor our blood pressure monitor also. All this comes under this. So ML integrated POCT, point of care platform for rapid diagnostics. So here this is there are 4 among them. Lateral flow immunoassay, vertical flow immunoassay, nucleic acid amplification test, and image sensors. So here lateral flow immunoassay, we all know the pregnancy kit, where we get 2 bands, or else in COVID-19 also, in the malaria test also, we have, they will be, when we put a minute amount of sample, what happens, the image, it will be powered with some AI, and we will get some the programming that was there in that. It will definitely give us in some bands, saying that if you get a 2 bands, it is an positive. If you get only single band, it is negative. Such kinds of assessment. Lateral flow immunoassay will give you your self-assessment and rapid test interpretation by using this machine learning algorithm. And vertical flow assay, it can help you to identify multiple biomarkers. In a cancer patient, they will be having multiple biomarkers. All the biomarkers can be quantified, or else can be identified by this vertical flow immunoassays. And the other one is nucleic acid amplification test. So this is used for rapid detection of viral and bacterial pathogen. And image sensors. So image sensors, they are used for to check any abnormalities, whether if it is in a cancer patient, any tissue or any cells or any organ, or else any biomarkers which are not needed that are present, that are immediately sensed by these image sensors, which are run by this deep learning analysis. So this is the image. Like you can see the lateral flow assay, where we all use, it is widely used. And even vertical flow assay, which was used to identify the biomarkers in the cancer patients. So they may be, if you want to do multiple biomarkers, quantify multiple biomarkers. So based on the algorithm that is mapped to this vertical flow assay, will totally quantify what all the biomarkers that are present in a blood sample. So very minute amount of blood sample is sufficient, or else the sample, any sample, even a urine sample, a very minute amount will give you a quick and robust result. So here it is nucleic acid amplification and image based sensors. Even certain certainly this will inject some chip into it, and based on that you can see whether what kind of tissue variation from the healthy tissue to the diseased tissue in cancer patients. If there is some variation, some biomarkers or else some variation in the tissue, all these imaging can be done. Some like I think days earlier, with the smartphone itself you can detect, the microscope, we have some microscopes that can be loaded into our phones, and based on that microscope itself you can identify certain pathogen, and you can give to a conclusion what kind of disease or what kind of pathogen is there in your blood sample or else urine sample. So next is how these biotechnology is involved highly, because you're all in this field. You might be looking into various opportunities. How this biotechnology is useful in this clinical application. So gene therapies. So early detection and treatment of genetic disorders like genome editing, as we have seen, and we can, if there is any genes that you want to mask or else any genes you want to treat it, so you can target certain microRNAs and you can mask, or else you can overexpress certain genes to treat that diseased condition or some disorders, and we can prepare some personalized medicines. So based on the person's lifestyle, based on the person's genetic profile, you can tell which treatment can be the person will be, so that we have seen that we all know that during cancer treatment so many people die because of the drug toxicity, that chemotherapies and all. So by this biotechnology, with all these techniques, you can design certain personalized medicine, avoiding this cytotoxicity into that person. And also for some immune diseases, you can also target some monoclonal antibodies, and even in cancer also you can address to some monoclonal antibodies, and you can treat the disease, and you can design certain vaccines also. As we have known, microRNA vaccine was also discovered, and also it was given. And also certain all these diagnostics, some cell therapy, suppose any damage, if there is damage of some liver, if there is a failure in the liver or any damage in the heart, so we can, by using this stem cell therapy, you can regenerate that tissue or else that organ, and you can do a transplantation to that person. And you can have an pharmacogenomics. By knowing the genetic makeup of that person, you can design your medicines, you can customize the medicines, and you can provide a good treatment to the people. So in the industry and in laboratory applications, so metabolic engineering, what is metabolic engineering? You can make some design some clones, and you can do an express certain certain proteins which are useful, which are very much needed for the society. By doing some genetic manipulation, you can do an metabolic engineering. You can get an enormous amount of certain proteins which are very needed. So by certain for diabetic medicine, the medicine of it is very costly. It says that it is extracted from the pancreas of the cow, so it is very expensive. So all these drugs which are used for diabetes, all those things can be produced by this biocatalyst. You can engineer some genes, and you can make it produce in a lab atmosphere. And also the synthetic biology and cell-free system is there. Without any organs, without any the cell organelles also, you can design your cell-free system by taking certain cell organelles from different, and you can assemble them together, and you can produce the proteins which are desired. So even in dairy industry also, it is widely used. This is and medical field also, to design the drugs and all these biotechnology techniques, all these techniques, drug designing, all these, and in the protein engineering, all these will be very helpful to address the challenges that the society is facing. Then laboratory applications, as we have all read about that. Multiomics in the sense like by giving your genomic, by taking the genomic and proteomic and metabolic profiling of any particular tissue, you can design the you can predict whether the candidate can be have a chance of probability of getting cancer or not, and you can edit the genes. You can you can delete and replace certain genes by using, as I have said, in CRISPR case. And also if there is an microfluidic chip, so what happens here, this chip, lung chip, when it is when it is inserted into the body, so it will it will monitor your breathing patterns also. And even so many cutting edge trends have come, like integrating AI and ML. So you can predict the protein structures also. By predicting the protein structures, by taking the amino acids, you can predict the protein structures. Once you predict the protein structures, you can design the targeted drug also. Which drug will bind? What are the binding sites that can be predicted from these protein structures also? You can assess by targeting. You can without the involvement of the lab techniques also, you can design your drugs and you can prescribe certain drugs. These are the targeted drugs that are to these sites. All these area, all these things will be at your edge. So my slides are visible?
Yes, ma'am.
So all these we have discussed how the portable imaging system, you can develop certain like ultrasound patches are available. Suppose if you want to measure certain bilirubin levels in the baby, in a little children, so you need not take the blood samples. Just by using certain scanners, handheld scanners, you can identify the bilirubin levels that are present in the liver. All these things can be designed. And as we explained, we can wear certain all these watches that we use, which will monitor our glucose, blood pressure, heart rates. All those things can be assessed, can be designed. We can address many diseases by these advanced biotechnology techniques. And also AI powered diagnostic system, as we have spoken, the machine learning and deep learning, they enhance the image analysis and reduce the diagnostic errors and streamline the clinical decision making for the better outcome. So that by all these techniques will make the redundancy will be reduced, and the patients will be given correct and right treatment without any side effects. So what are the career opportunities with this field? In the field of diagnostics, what are the career opportunities? Not only in the diagnostics, actually this biotechnology field itself is a robust, and it has a wide range of opportunities that we can walk through based on our interest, based on our expertise. So like there are so many opportunities who have studied from this field. They can work as a research scientist in analyzing designing the experiments and doing some and solving some biological problems, even bioinformatics scientist, clinical research coordinate, by working in the industry for developing any biotechnology product. You can work as a regulatory affair managers, quality control specialist, industrial technician, microbiologist, biomanufacturing engineer, process development scientist, and so many opportunities are there who are very much interested in this field. So there are a wide range of based on research, and also in some clinicals there are a huge area of opportunities that person can. So what should we do to get a good career, to have a successful career? What should be our strategy to develop our skills, to develop our talents, or else our our expertise? How should we develop it? How should we nurture ourselves to be a successful candidate in this field? So we have to advance our degrees anyway, that is must. And we have to, and as a MSc students, you should be always working with some analytics. You should be you should have a good calculation skills, and you should be always coming forward to troubleshoot any problems that you face through, or else any challenges, means in the laboratory, in the wet lab, any challenges you have facing, you should try to troubleshoot it, and you should optimize. You should have your own protocols. So any protocols, you should try to standardize it, and you should be, your hand should be very handy to doing the lab techniques, and also you should have a very good data analysis skills, and you should be very you should be always discussing. You should have a good peer group, like good communicating skills, and you should be in an what to say, you should have a good relationships, like with the companies, with good networking, in the sense I need to say. And you should have, and you should be all the lab techniques that are, which are happening with your within your around, within your nearby laboratories. You should be have an idea what all the techniques that laboratory work upon, what all the hypothesis, or what is the area those laboratories working about, whether all those things, the areas that they are working, you should try to discuss with them, and you should try to expertise, and you should have an hands-on experience upon all these techniques, like cell culture, like CRISPR. If any laboratory is working on the CRISPR, you need to discuss. If you have any hypothesis with them, whether to check whether it is, and whether your hypothesis is correct, can it be implemented or not, all those things you should be always be mastered in these techniques. And also bioinformatics, also, like bioinformatics is everyone is nowadays exposed to this, and it is a very without this nowadays we cannot go ahead in our career life. Like you should know this some minimum software skills, and also you should be always be attending the certification courses, some conferences, and during your break, winter break or summer break, you should take an opportunity to attend some internships. And also you should you should know the ethical factors that all should be used in the laboratories. What all the regulatory knowledge, how to, if you want to do an animal experiment, how should you approach further, how should you to take the animal ethics, how should you follow the animal ethics, what are the CCSA guidelines, what are the FDA guidelines. If you want to do some protein engineering, whether you want to develop any dairy product, what is the protocol that you have to follow to get an FDA approval. All this minimum knowledge you should be having, and you should be well versed. You should have an idea by yourself to write some grants also, and you should have, suppose if you want to generate some product, and if you want to get a patent, all these things, like having some IPR knowledge also should be very much needed to succeed in this field. Thank you.
Ma'am, you have really explained very well all the aspects of the applied biology, how they can use the different techniques and other things in their knowledge. And it has really been a good expertise lecture for them. And I think they have been benefited, and they will be benefited. They will utilize all these things in their further studies. Here there is a mix of graduate and postgraduate students. So graduation students will definitely learn all these things in their post-graduation, and the post-graduation students will apply all these knowledges that they are getting from all the lectures in their further studies. So thank you so much. And Yash, please give a formal thanks to ma'am, and then I will connect over to you, ma'am, on call.
Yeah.
Oh, yes. Over to you, Yash.
Okay, ma'am.
Please ask them to clarify it here only.
And anyone is having any doubt, any query, you can ask.
If there will be any question, ma'am, we will definitely drop you a mail. It's because I think the students must be trying to understand what they should ask, because immediately these are some complicated terminologies for them. I think at this point of time, they will definitely drop some questions, and then we will forward it to you for better explanation.
Okay, okay. Thank you, Pratima. Thank you,
Yes,
Ed, for giving this opportunity. So I really enjoyed. Hope you also enjoyed my lecture.
we
And
enjoyed, ma'am. We enjoyed. Yes,
Okay,
please
so
drop a formal.
as we come to the conclusion of this enriching expert lecture on biotechnology and diagnostics, on behalf of Kaustav Careerica and Prakriti Private Limited, I would like to express our deepest gratitude to our distinguished speaker, Dr. N. Veena Rani, for delivering such an insightful, intellectually stimulating, and impactful session. Her comprehensive discussion on translational applications of molecular approaches truly reflected the depth of her expertise and research excellence. The way she seamlessly connected fundamental science with the real world diagnostic innovation has been immensely valuable for all of us. We are sincerely thankful to you, ma'am, for taking out your valuable time for your demanding research schedule and for sharing your knowledge, experience, and research journey with our participants. Your lecture has not only enhanced conceptual clarity but has also inspired many young minds to pursue meaningful research in diagnostics and translational biotechnology.
thank you, Ash. Thank you so much. Looking forward for your questions, and we will stay connected. So whenever you need, I will there for you to give some lectures. And I
Yeah,
hope
ma'am.
even Kaustav.
We will forward the questions if they
Yeah,
will have.
yeah, definitely, definitely. I will answer. I will be very fortunate to get answered them. So, thank you so much, Pratima. Thank you, S, and thank you for the team, Kaustav, Careerica. So, thanks for giving this opportunity, and we'll stay connected. Anytime you need, I'm there for you. So, I will extend my time, and I will definitely be a part of your team. Thank you.
Thank you
Thank
so
you so
much,
much,
ma'am.
ma'am.
Thank you so much. We will definitely work on long-term collaborations, and we'll see how we can collaborate further. Thank you so much for your valuable time.
Thank you. Bye. Good night.
Good night, ma'am. Students, hello, all of you are there?
Yes, ma'am. Yes, ma'am.
Yes, ma'am.
अरे एक यश मैम बोल रहा है, गौतम is there. Others, are you there?
दो ही बच्चे हैं, सब भाग गए।
सब भाग गए?
हाँ,
Don't say
दो ही
this
बच्चे
here.
हैं।
Don't say this here. Okay, no issues. You people, I have uploaded certain videos from my own expert lectures and other things on the LMS. You people can go through it, and then I have assigned it to every